Okay, let's learn a little bit about, what changes things from resting potential. So what we're on right now are the forces. So we learned from the last module about what the neuron is called at rest. It's polarized, it's negative 70 charge. The reason why it's negative 70 charge is because there's these large protein ions trapped on the inside. But now, how would we get it from resting potential to an action potential? So what you have is ions, and there's two main forces that we're working with, the concentration gradient. And the concentration gradient is ions tend to flow from higher concentrations to lower concentrations. So if you could picture like uh, let's say a square box, and it's an acrylic box, and it's got some salt in it, and the salt comes up to a certain level. But then I pull a side of the box out. What would the salt do? It would flow to an area of lower concentration. So you understand that is that ions tend to flow from higher concentrations to lower concentrations. If you were on an elevator and the door opened and you felt that it was too crowded, you go to an area where there's less people. If I open up a few more desks in the room, then people will tend to spread out instead of staying in just one side of the room. So you can think of all these examples like ions as they tend to flow from higher concentrations to lower concentrations. Then the electrical gradient is one that a lot of you know about. Opposites attract. That's not really true with humans. We do better with similar, but it's okay. When you think about opposites, just a little bit of differences seems to make things work. But we're talking about ions here. So electrical gradient, opposites attract. So the positives are attracted to the negatives, and so on and so forth. So I made this little chart here, and I thought I could show you how we could do this together. So let's think of the resting potential, okay? We're back to this resting potential. So here's our little axon and the dendrites, right? And here's the nucleus, okay? So we know that inside during resting potential it's negative 70 millivolts, okay? And then we know that K is trapped on the inside, and we know that Na is more concentrated on the outside. So let's look at the concentration gradient. There's more sodium here than in here, so what is it going to want to do? It's want to go in, right? Go in. And then the electrical gradient, is the positive attracted to the negative? Yes, it is. So it says, I want to go in too. Okay, so you've got two forces just pulling sodium in. Oh, we want to get in, we want to get in. But remember, you have this semi-permeable membrane that's saying, no, you have to stay out. We didn't let you in. Okay, so now let's look at potassium. Concentration gradient. Yeah, there's a lot more potassium here than out here, so it kind of wants to go out. Go out. Okay. But then let's see what happens with it. The electrical gradient, K, does it want to go out or does it want to stay in? It wants to go in. So K is kind of being wishy-washy. A part of it wants to go out and the other part wants to stay in. Do any of you feel like that? Oh well, we won't go there. I can't talk to you anyway. Okay, so here we go. Let's go on to the next slide. Okay, so what you've got is you've got two forces pulling Na in, and then K is sometimes going out, sometimes staying in. It seems to be kind of wishy-washy. So what keeps it at negative 70? Let's see our friend. Oh, no, we're not getting to our friend yet. We're going to depolarization. Okay. Depolarization is when the inside of the cell becomes more positive. So since that Na wants to get into the cell and it's got two forces pushing it in, if you remember, it's a semi-permeable membrane, so some of them can slip through. What I do in class when I have a face-to-face is I will usually bring 2 bottles of solution, clear glasses. And in this one I put sugar. Okay, so this one is sugar. And then this one is pepper. And there's water in there. Okay, so can you imagine pepper with water and sugar with water? So if I mix the sugar up really well, will I see the sugar in the water? Nope. But if I mix the pepper up in the water, will I still see the pepper in the water? Yes, I will. So then I have this nice white sponge. Okay, this is the sponge, and this would represent the semi-permeable membrane. So when I dunk this in here and I lift it up, we don't see anything. Probably the sugar can go through. That's like the potassium. The sugar's representing the potassium, as it can go through pretty easily since this is semi-permeable. But when I put the sponge in with the pepper is trapped on the outside, and that's more the Na. But then I cut it in half, and is there some pepper that gets through? Yes, there is some pepper that gets through. And so that's what happens here, is sodium can kind of slip through. And why would it slip through? Because it's got two forces pushing on it. So when the inside of the cell goes from like negative 70, which is its resting potential, to negative 68, to negative 66, to negative 64, to negative 50, anything that's less negative, more positive, less negative, then we say that it's depolarizing. Depolarization is when the inside of the cell becomes more positive. Okay, let's go to our next one. Okay, so this was the friend that I was telling you about. What we have is we have something inside the cell. It's a protein molecule located on the inside of the cell that attempts to maintain the resting potential of the cell. So what you have is, I'm going to show you these videos, and you're going to see that this thing right here is the sodium-potassium pump, and what it does is I call it the equalizer of the cell. It's like the bouncer in the cell that says, you know what? K is supposed to be on the inside, but Na is not supposed to be on the inside. So what it does is it does its job of going ahead and it will pump K into the cell and pump Na out of the cell. And what it does is it tries to maintain the homeostasis of the cell, or the balance of the cell. So I have two videos here, and I'm going to play them for you. But what I have to do when I'm playing the videos is I have to stop the microphone so I can't tell you, go in between, and then I put this one on. So this one here is just a cute little video of kids. I don't know, maybe I'll come back and tell you, oh, this is a cute video. We'll see. Okay, so let's go watch this video now.
Animal cells store energy in the form of ion gradients across the cell membrane. In the cytosol, the sodium ion concentration is kept low relative to the extracellular fluid, and conversely, the potassium ion concentration in the cytosol is kept high. Like water behind a dam, these gradients harbor potential energy that the cell taps to fuel cellular work. Animal cells use a membrane pump, called the sodium-potassium pump, to maintain these ion gradients. To begin the pumping cycle, sodium ions enter binding sites on the cytosolic side of the pump. Although there are 3 sodium binding sites on this pump, for simplicity only one is illustrated here. Pumping sodium against its concentration gradient requires energy, which is provided by cleaving ATP. ATP transfers a phosphate group to the pump in a high-energy linkage. Phosphorylation causes a dramatic change in the pump's conformation, so that the sodium ions become exposed and released outside of the cell. This action also exposes binding sites for potassium ions in the pump. Although there are 2 potassium binding sites, for simplicity only one is shown here. Binding of the potassium ions triggers release of the phosphate group and the return of the pump to its initial conformation. The potassium is then released inside the cell, and the cycle repeats. A complete cycle takes about 10 milliseconds.
Okay, so you saw a video animation of the sodium potassium pump. Now I found this video online and I thought it was cute, so I'll play it for you twice. It's like little kids that are acting out the sodium potassium pump.